First, I'd like to thank all the organizers for all of their hard work and for the opportunity to be here. It's been tremendous. So I'm going to talk today about a brand new study that we're currently working on in Marlene Cohen's lab that addresses how attention modulates population activity in macaque visual cortex. And as you can see from my title, and despite Alex's career advice yesterday, I will be talking about noise correlations. Before I start, I want to acknowledge Regina Chang, who's the tech in the lab, who's collaborated on this project with me. And when I say we, I'm referring specifically to her. One of the focuses of the Cohen lab is to study population activity in sensory cortex and understand how this activity relates to perception and behavior. And in addition to this, I'm particularly interested in studying how the internal state of an animal, particularly attention, affects perception and behavior. And so in the lab, we like to study visual attention for two main reasons. And the first is that both humans and monkeys are pretty good at directing their attention to locations or objects. And the second is that it's well controlled. Attention is well controlled experimentally. And to make this point, briefly regarding this first point, here's Lionel Messi lining up to take a penalty shot for FC Barcelona. And as he runs up to take his shot, you see that he's looking directly at the ball, but he's also covertly attending to where the goalkeeper is and where he's aiming his shot. And if he looks directly where he's going to aim his shot, he'll tip off to the goalkeeper where he's shooting, and his shot might get blocked. But of course, that won't happen, right? And so we call what he's doing covert spatial attention. And he made the shot, and it turns out that monkeys are also very good at covertly attending. And what I'm showing here is this smaller monkey is looking away from his larger companion who's threatening him. And in monkey society, direct eye contact can be perceived as a threat. And so you can guarantee that while this smaller monkey isn't making direct eye contact with this larger guy, he's definitely attending to what this larger guy is doing, right? And so in the lab, we don't bring in big monkeys to scare our smaller monkeys to get them to attend. But instead what we do is we offer them rewards to pay attention to a particular location in space. And when we do this, we can then quantitatively assess the effects of, the perceptual effects of attention by collecting psychophysical data. And in the interest of time, you guys have all seen behavior like this before. But the basic point is that you can cue an animal to do a perceptual task at a certain location. And when you do that, you can get these red psychometric curves. And on some subset of trials, you can trick the animal and have whatever task the animal is doing, in this case a detection task, you can have the thing that the animal is supposed to detect happen at a different location. And when you do that, you can see relatively large differences in behavior. And this is quantifiable, and it's readily quantifiable. And so oftentimes people talk about this difference here as being the magnitude of an attentional effect. Sometimes people talk about the shift in the psychometric curve. But either way, it's clear from these data that attention allows us to see better. And what we really want to understand is, how does attention do that? And there's been a lot of work in the last 20 plus years that's shown that attention directed to a particular location in space increases the firing rate of sensory neurons that represent that location in space. And recently, there have been some observations, including the talk that we just saw, the excellent talk that we just saw, that have shown that attention can decrease correlations amongst pairs of neurons. And I think that the previous talk is the first inkling we have into a mechanism about how that might happen. And so by noise correlations, we all know by this point in the meeting, but I specifically mean the trial-by-trial covariation in the firing rate of a pair of neurons. And so several groups now have found that there's this inverse relationship between firing rate increases due to attention and changes in noise correlation. And I'm showing a figure here from Cohen and Mansell in 2011 that shows that both spatial and feature attention have similar properties here, that as attention increases the firing rates of pairs of neurons, it decreases their correlation. And one of the things that I was interested in when I saw these data is whether this relationship between increased firing rates and attention and decreased correlations is fixed, or whether this relationship is flexible. And so it could be that biologically, firing rate increases due to attention are always accompanied by decreases in correlation. And coupled with that possibility is the idea that attention isn't actually changing correlations on purpose. It's just sort of along for the ride, right? And maybe there's some people in here that think that. And alternatively, what might be happening is that attention does whatever's best for information coding. And it may be able to flexibly alter noise correlations depending on the experimental context. So in the study I'll talk about today, we asked the question about whether attention can flexibly alter noise correlations amongst pairs of neurons. And to really nail this, our goal is going to be to basically try to decouple rate and correlation changes that are associated with attention. And so as we thought about this issue, it became clear to us that previous studies looking at attention and correlations had all used tasks where nearly all the neurons that were recorded from were contributed to the same behavioral choice. So these were typically detection tasks. And if I need to go into the details of explaining what I mean, please interrupt me. I'll try to make it more clear. But the point here really is that while we don't have an idea exactly of the role of correlations on readout, and this is complicated, and as this meeting has made clear, it's far from resolved, when neurons are all contributing to the same behavioral task, their activity might simply be averaged together in readout. And in a case where that happens, reducing correlations could conceivably increase the sensitivity of a population's response. And so this cartoon, this walkthrough may be unnecessary for this audience, so I'll try to go quickly through it. But let's say a monkey has to make a judgment about this Gabor patch, its presence or absence, what its contrast is. So we know that a large proportion of neurons in visual cortex respond more to stimuli with higher contrast. And so a simple thing the brain might want to do is average the activity of visual neurons that respond to this stimulus to determine what the contrast of this patch is, whether it's present or not. And I'm skipping ahead here, sorry. And so what I'm showing in this example here is are 6 example neurons, 6 simulated neurons that have independent noise. And when you average, the x-axis here is trial numbers, repeated presentations of the same stimulus. And what you see is when you average the activity of these 6 neurons together, you get a relatively stable estimate of what the mean is across trials. And so basically averaging these neurons together reduces the independent variability. However, in this example on the right, these neurons all have correlated variability. And so when you, obviously when you average these together, this correlated noise doesn't average out. And so we think that this type of averaging might have been what's going on in these previous studies that have looked at attention and correlations. And so in those cases, maybe decreasing noise correlations is advantageous for population sensitivity, right? But what if instead you wanted to tell which of a pair of Gabor patches is of a higher contrast? So there are 2 stimuli now. So a simple solution might be to first average the responses of neurons that respond to stimulus 1, here group 1, and then average the responses of neurons that respond to group 2, and then compare the activity of those 2 groups, maybe by a subtraction here, right? And you can see here that when there's correlated variability between groups, when you do the subtraction, you remove that variability, and you have a more stable estimate than when the variability is independent. So in this case, maybe increasing correlations between groups might be advantageous. And I guess another way of talking about this is saying that if you're going to have variability in the brain, in this particular situation, it would be good if that variability is shared between the groups, and so that it can be subtracted out. So with this idea in mind, we designed a task to ask whether attention modulates correlations differently when neurons contribute information in support of the same or different choices. And for the purposes here, we've decided to use a contrast discrimination task, and where the monkey has to look at these 2 stimuli and tell us which is of a higher contrast. And so with this task, there are going to be some neurons that respond more to stimulus 1 and some that respond more to stimulus 2. And ideally, after averaging the responses of neurons that represent each stimulus, the brain should compare the activity of the neurons in each group to figure out which stimulus is of a higher contrast. And so because we want to ask what attention does to correlations, we're going to actually have 2 pairs of stimuli in every trial. And we're going to cue the animal to attend to one set of stimuli at a time. And what we want to know is what's going to happen to the correlations between pairs of neurons that are in different groups, that represent different stimuli when the animal attends versus when the animal doesn't attend to these stimuli. So let me briefly walk through the schematic of our task. First, the animal maintains fixation, and then after a random period of time, these 4 stimuli show up. I hope everyone can see these. And they were on for 4 different sets of durations that range between 400 and 1,000 milliseconds. And we did that for purposes that I won't talk about today, but it was essentially to look at the effects of different durations of time for integration. And so what we did is we cued the animal in blocks to attend to one set of stimuli or the other. And the animal always has to make a discrimination based on the 2 stimuli that are on the same side. And I'll explain how we cued the animal in a moment. And then after the stimuli were up for the randomly selected period of time, the fixation spot extinguished, the stimuli went away, and at the same time, 2 targets went up at the attended locations, and the animal's job was to saccade to whichever stimulus was of a higher contrast. So to make sure that the animal wasn't ever using these unattended stimuli to help his judgment, we counterbalanced everything so that he couldn't use these stimuli to compare against these. So the unattended stimuli could be of a higher contrast, of the same contrast, of a lower contrast as the correct answer on the attended side. And just to show that that's the way it worked out, this is a behavior from an example day where the animal's overall percent correct was about 70%. And then if we sort his correct and incorrect trials as a function of the unattended contrasts, whether they were higher or lower than the correct answer, or whether they were the same pair or a different pair than the same answer, you can see that these unattended stimuli had no effect on behavior, which suggests that the animal was attending properly and basing his judgment off of the correct 2 stimuli. And so in addition to the main task that I just described, we also intermixed throughout an experimental session instruction trials. And on these trials, the temporal structure was the same, but only 1 stimulus appeared. And the animal's job was to saccade to the target that signified the location of where the 1 stimulus appeared. And we had these at the beginning of blocks and also throughout blocks. And these stimuli could be at any of the contrasts that were used during the main experiment or also at full contrast. And we use these to cue the animal to what side to attend to. And also, as I'll explain in a few moments, we use data collected from these trials to assess what pool a neuron belonged in. And I'll explain exactly what I mean by that in a minute. Before I get into that, though, we recorded from Cortex by making chronic array recordings using Utah arrays. And we implanted 2 of these arrays, like this one, one each in bilateral V4. And I think most of you know that V4 is a mid-level visual area in the ventral processing stream, and previous work has shown that these neurons are modulated by attention, and also that attention decreases their correlations, right? And so here I'm showing example receptive fields from a previous study using arrays like this in V4. And what we're doing for this experiment is we're putting our Gabor patches within the envelope of the receptive fields of the neurons on each array, right? And the particular array that we used contained 48 electrodes. And so on a single day, we recorded just under 100 neurons total. And by neurons, I mean both single and sorted multi-units. During the task, our average firing rate was about 16 spikes per second. And we counted spikes over 400 millisecond windows, starting with stimulus onset. And so because we can't say for certain with these types of recordings whether we're looking at the same neurons from day to day, we now like to take a conservative approach and look for the effects that I'll describe today within a single day's recording session. And so we're going to look for these effects within a single day and then look for the effects across days. And so I'm going to show our results on the following axes. On the y-axis, I'll show you spike count correlation. And on the x-axis, I'm going to show you this measure that we're calling test tuning similarity. And we think that this is, what this is we take the product of d primes for a pair of neurons. And I'll explain how we calculate this right now. So we're still trying to figure out exactly what we should call this. But basically, it's related to the location of the receptive fields of pairs of neurons. But specifically, it's a measure of whether a pair of neurons commonly respond more to the same stimulus or not. So we can think of it as asking whether a pair of neurons contributes information for the same or different choices in the context of this task. And we like to use this because we're simultaneously recording from lots of neurons, and so we can't optimize where our stimuli are for all the neurons that are. So we think that this gives us leverage on our question. And so to calculate this, we take a two-step approach. And the first thing that we do is we use the responses from these instruction trials to full contrast stimuli. And then we calculate a D prime value for each neuron, for its responses to stimulus 1 and stimulus 2. And when we do this calculation, we subtract the responses to stimulus 2 from the responses to stimulus 1, so that when a neuron has a positive value, it means it responds more to stimulus 1. When a neuron has a negative value, it responds more to stimulus 2. And just to make this clear, it's hard to see these colors, but here's a cartoon of 3 example neurons. Neuron A and B have similar receptive fields, and they overlap, and stimulus 1 is contained within the extent of their receptive field. Neuron C here, maybe you can faintly make out, its receptive field has stimulus 2 in it. So neurons A and B are going to have positive D primes because they're going to respond more to stimulus 1. And neuron C is going to have a negative D prime because it responds more to stimulus 2. And so when we take the product of each pair's D prime, neurons A and B are going to have a positive product, and they'll be right here or somewhere in this range. And we take that to mean that these neurons represent the information about the same stimulus and contribute to the same choice, right? But neuron C has a negative D prime, so the products of neurons A and C and B and C will be negative. And we take this to mean that these neurons represent information for different choices. Right? So our first prediction here is that correlations are going to depend on this task tuning similarity measure. So we think that they're going to increase with increasing similarity. And that's going to look something like this on these axes. And so this says that neurons with more similar tuning are going to have higher noise correlations. And that's something that's been observed previously and has been discussed several times during this meeting, right? So the question then is, what's attention going to do to these correlations? And so the first possibility here is that attention is going to always reduce correlations. So it's going to decrease correlations across the board when the animal attends to the pair of stimuli for the discrimination, right? The second possibility is that attention does what's best for information coding. And so in this case, it means that attention will decrease correlations amongst neurons that are in the same pool, but it'll increase correlations amongst neurons that are in different pools. And so I guess it's worth saying again that over here on the left, this is where shared variability will be subtracted out between groups. Right? And so I guess it's also worth pointing out that both of these hypotheses make the same prediction for neurons that are in the same pool. And the previous studies that have looked at correlations and attention have only looked at neurons that are in the same pool, we argue. But the difference here and the novelty of this design is that it allows us to ask what happens between neurons in different pools. And here there's a distinct opposite prediction, right? So what have we actually found? Well first of all, we found that these are results from a single day of data from our first monkey. And we've completed a full data set of collection from one monkey, and this week we've started recording from our second animal. And so what we see is that noise correlations do seem to depend on task tuning similarity, such that neurons representing the same choice have higher noise correlations, and neurons representing opposite choices have lower noise correlations. And regarding the range and magnitude of these are slightly larger than Marlene has observed previously, but not completely out of the realm of possibilities. From some of the animals she recorded from in grad school, apparently. I have limited experience with different animals and noise correlations. But these are also longer windows than in her previous study. So these are 400 millisecond windows. So what we found then with attention is that, again, attention decreases correlation amongst pairs of neurons that are in the same pool. And in confirmation of hypothesis 2, it increases correlations between pairs of neurons that represent opposite choices. And it turns out that this finding is extremely robust. We've actually found it on every single day of data collection that we've done where we have sufficient data. And so another thing to remind you of this, at this point is that because we're using bilateral arrays, the behavior on this green line and the red line are exactly identical. They're collected at the same exact time. So there are no differences in uncontrolled cognitive state differences between these two lines, right? So the critical observation here is that we've demonstrated a decoupling between firing rate changes and correlation changes with attention. And attention increases firing rates for this green line, but the direction of the direction of changes in correlation depends on which pool these neurons are in. So briefly, I just want to summarize these data. And so what we've done here is taken cell pairs where the D prime was less than -1 or greater than -1, and just collapsed and averaged to show the correlation values. So this is a summary of what you just saw. And then at this stage, we have two basic concerns about the results. The first is that we know that there's a well-documented relationship between firing rates and correlations. And so we wanted to look at matched firing rate distributions amongst these pairs to make sure that these results hold. And then the second is that these two sides, these two pools, differ in terms of their receptive field similarity. So there's also a well-documented difference in the baseline correlation rates between neurons that have different receptive fields. So we want to look to see if that's an issue with these data as well. So first what we did is we took these data, so these raw data with greater than 1 D prime products and less than -1. And we subsampled them, we mean matched them so that all 4 bins of data here and here have the same distribution of firing rates. And again, we found the same pattern of correlation changes for these pairs, so even though the firing rates were matched. And then likewise, we subsampled, oh, those colors came out weird. We subsampled the match for correlations in the attended condition between the same and opposite choice pairs. So for these bars, this and this, by definition, these correlation values are identical. And then we asked, what happens to the unattended correlations? And what we see is the same pattern here. So it's not due to differences in baseline correlation levels, right? So I'll conclude here, and what we've shown here is that the relationship between increased firing rates and decreased noise correlations is not fixed, and that instead attention flexibly modulates population activity in a way that's best for information coding. And we can say that because attention decreased correlations between neurons in the same group, and it increased correlations between neurons in the opposite group. And so our results suggest that attention works on rates and correlations separately. And the talk previously maybe gave interesting insight into a mechanism about how that might work. But we have currently no other strong hypotheses about how that might happen. So I think that's all I'll show for now. So thanks very much.
So I have a question with respect to your tuning similarity. I'm not quite sure I fully understand it. So what I deduced, it seems to be basically a spatial similarity. Is that right?
It's not entirely. It's within the context of this task. What's the similarity? How do neurons respond? Do they respond similarly or differently to these two stimuli? So a neuron could encompass. Sorry, I'm going the wrong way here. A neuron could respond equally to both stimuli, and in that case, it would have a 0. It would fall on a 0 here, right? But if a neuron responds very strongly to one stimulus,
so, but two stimuli means you present the stimuli individually in those locations, yes, and you match the contrast, of course.
Yes. So what we do in those instruction trials is we show stimuli at the 4 locations of the task, at the contrast used, but also at full contrast.
But given that this is a contrast discrimination task, why don't you simply sort your neurons according to where their contrast thresholds or C50s, for example, are?
That would be another way to do it. I think that would be a reasonable thing to try. We haven't done that yet. We like this because what it asks within the context of this task is how much information these neurons have in support of one choice over another. And we think that this captures that question fairly well.
So you have pattern recording, so how about just like decoding and
condition
that you tried?
Yeah, we're certainly not allergic to that idea. We like that idea. These data are very new and I haven't done that yet, but we plan to.
I just want to double check about the
D prime.
The tuning index.
Yes.
It was calculated from the presentations of both. I wasn't sure when you said you do the single stimulus presentation. You're not using the single stimulus presentation to calculate the tuning index.
Yes, we are. So we're using the situation here. So we want to know.
So why don't you just use the real data?
So that's
making strong assumptions.
yes,
about surroundings.
exactly. So that's one issue. So one thing that we still need to do is use some subset of trials to make this so parcel out some set of trials and do a similar calculation, a similar index, and then use the remaining trials to look for noise correlation. And we can do that. We just haven't done it.
How far from threshold are you? I mean, you know, dynamic range in those cases, of course, say physically, but
Yeah.
also.
So this is what we do is we've trained this animal a long time and we have a sense of its performance, and what we aim for is roughly about 70% correct. And so we have both discrimination, we have all the sets of discriminations are differences.
Neurons, my memory, I think it was maybe 4 years ago, what the monkey can do and the neurons can do is very, very different.
Sure.
Neurons really can't do much in terms of much higher contrast thresholds than the monkey itself. I'm just wondering what the dynamic range is before you actually run, because you have to be at higher contrast when the monkey is really closer to.
Yeah, I think
70%.
70% correct.
Yeah, I wish you would have to be at very low contrast.
Well, it just depends what the relationship
Yeah.
between the contrasts are, right? So you know, there's a known relationship between
You don't think it's important? I just was wondering.
I think for these
Dynamic
questions,
range or
I think for these questions, it's not particularly important. But there are other things that we can ask that would be better served by more difficult discriminations. I certainly.
So maybe just another thing, but you seem to say that it's possible that attention increases firing rates but decreases correlations. Did you show us firing rate changes?
I did not show firing rate changes here. We see this typical sort of thing with attention. I think in this animal, the average firing rate increase with spatial attention was about 70%.
So the firing rate is increased for all neurons, regardless of the.
Well, for neurons that are that have receptor fields on the attended side,
Yes,
right?
yes.
Yes.
So right now, I'm still a bit confused about
Yeah, okay.
the product G primes and exactly what it means, because it's not really signal correlations, it's something else. Have you done a sanity check, just like running a simple model where you take cells with little properties, you simulate them, you put the kind of correlation patterns you think are there, you compute the same kind of similarity index, and then show that your results would indeed imply information increase?
No, we haven't done exactly that yet. No. We've done simpler versions of that in anticipation of this project, of doing this project to test that our predictions, that the two hypotheses were not, that one was not a straw man, I guess to say. But specifically what you suggest is worth doing, and we'll do that. So it sounds like I didn't explain this product of D primes very well. Should I go into this more? Or
For me, it was very clear what it was.
Okay.
What's unclear is exactly how it relates to the correlation and to information.
Sure, yeah. So we don't want to just ask what's the amount of receptive field overlap in all of these neurons, because that's a question that's not necessarily related to the specifics of this task, which is what neurons provide information in support of this stimulus versus what neurons provide information in support of this stimulus. So if a neuron responds equally to both stimuli, it's going to have a 0 for its D prime, and it's not going to contribute to in a pair on our plots here. It's going to fall. It's going to fall here with a pair of other neurons. But if both neurons strongly differentiate between stimulus 1, respond strongly to stimulus 1 and less strongly to stimulus 2, they'll be over here. And so we think that they're both providing evidence in support of the same choice.
The G prime was computed for one particular contrast value, or your average across
So
all
we
contrasts?
use full.
You could imagine all sorts of situations
Absolutely.
where
And that's-
in fact, they're the same for one contrast, but not for other contrasts, and you can tell the difference.
Absolutely, and that's something that we need to look at. So we have all of those data with different contrasts, and then we have all of the data obviously during the task. And so what we can do is calculate these values at lower contrasts and look for the same thing, and we can calculate them using a subset of the task data specifically, so that we can account for surround effects, and we will do that.
Did you look at these results also if you separate neurons by preferred orientation, or how the orientation relates to the orientation of this contrast?
We've not looked at that. What our sense from looking at the data is that the orientation tuning of these cells doesn't have a major impact in this task. That most of the V4 neurons do show, as we would expect, increasing contrast response functions, regardless of what their orientation preference is when we show these stimuli. And what we know from Marlene's previous study, which was a flashing change, orientation change detection task, is that all of the neurons almost all of the neurons that she showed, probably due to adaptation, showed an increased response to the change stimuli, regardless of what their orientation tuning was. And so, in that context, it didn't seem to matter much. And I also think that in this context, it won't matter much. Saved by the bell here, huh?
Yeah, I think I want to keep on schedule, so if we move on, we can always ask this question later. I won't ask mine, you won't ask the other.